Good morning, everyone. Welcome to Saturday Physics. This is the last lecture of this fall. We'll be back next year. So if you have any suggestions or comments, you can talk to us or write to us. So today it's my pleasure to introduce you Professor Seppe Kuehn. Professor Kuehn received his PhD in chemical physics from Cornell University. Then he decided biology is more exciting. After a fellowship at Rockefeller University, Professor Kuehn moved here in 2014, and his group uses microbial systems to study the complex ecological and biological dynamics in the natural world. And his research has led to some surprising insights into the adaptive evolution. Let's welcome Professor Kuehn.
All right, welcome. Can you hear me? Yes? It's been a few semesters since I taught 212 in this room, but thanks so much for coming to Saturday Physics. It's going to be a little bit different today. We're going to talk about evolutionary dynamics and not magnets or stars or anything like that. So I'd just like to start with a story. In the early 1850s, a guy named Alfred Russel Wallace was in the ocean around Malaysia on one of these British fact-finding missions. And he was out there, of course, like collecting organisms and plants. He was a naturalist. And he got malaria. And he'd recently read a book by Malthus about how populations were going to explode. And his claim, at least, was that during one of these feverish bouts of malaria, he had a vision which was essentially the idea of evolution by natural selection. And so he realized that this rapid population growth, coupled with variation and inheritance, could give you a process which could generate new species and cause life to diversify. And so he very quickly wrote a letter to George Lyell, who was the head of the Royal Society in London. This is the biggest, most important scientist in the world at the time, back when Britain wasn't doing Brexit, but was doing global expansion. Lyell got this letter, and he took it to his friend Darwin and said, "Hey, Darwin, the idea you've been working on for 25 years, somebody else has it now." And Darwin freaks out, and Lyell freaks out because he doesn't want his friend Darwin to be scooped by Wallace, who's basically had the same idea. So Darwin and Wallace agree to write a letter to the Royal Society. They write a letter to the Royal Society. They have a meeting. They give this talk about the evolution by natural selection. It's like the most important idea ever. Nobody understands it. And everybody in the Royal Society is sort of like, ho-hum, let's go on to the next idea. Nobody's interested. So then Darwin writes a short summary of his 25 years of work, and that short summary is a 300-page book called The Origin of Species. And that 300-page book contains this passage, which is sort of towards the end of Darwin working all of this stuff out. And he's, you have to understand that Darwin is arguing for a whole different way to think about the origin of life on Earth, right? Everybody thinks God is responsible, and he's saying that there's this law that's operating that can give rise to this, to life. And so he's a bit under the gun, right? He knows what happened to Galileo. It didn't turn out well. He's got to make a good argument, and so the prose in this book is truly remarkable, not only the science, but the prose. So I want to read this passage to you, which is really remarkable. So he evokes this image of a tangled bank, and he says, "It's interesting to contemplate the tangled bank clothed in many plants of many kinds, with birds singing in bushes and various insects flitting about." He's a real naturalist, right? This guy studied sponges in Scotland for 10 years. And with worms crawling through the damp earth, and reflect on these elaborately constructed forms that have all been produced by laws acting around us. So when I read this passage, I think Darwin was a physicist, because Darwin had really uncovered laws that structured the natural environment, the living matter that we see on Earth. And so the question that we're trying to answer in biological physics is, what are those laws? He gives us a sketch, but we're not quite there. So that's one of the things that we're trying to do in my group. So what are the laws that Darwin, and to be fair, Wallace really had the same idea. We always think about Darwin, but I would like to give Wallace a bit more credit. So what is evolution according to these two folks? There's basically three things which you all probably know from thinking about evolution in high school. And those three things are variation, That is, things need to change. They need to have adaptive capacity. They need variation in their genetics or in their phenotypes. Those are the outward expression of the genes. They need to be able to transmit that. You've got to communicate it to your kids, because if it works well, they ought to use it as well, right? So there has to be transmission, and there also has to be competition. And competition is the process by which you have different variants that compete. This is Malthus, and the best one wins, and then natural selection proceeds. So these are the three main ingredients. Competition is sometimes called the struggle for existence. That was the chapter heading in Darwin's book. And so these are the ingredients that are a necessary element for evolution by natural selection. And what I'm going to talk about today is the first one. What do we understand about how variation happens? What do we understand about really the constraints on variation? Can organisms just do whatever they want, or are there bounds? And it turns out there are bounds, and that's basically the topic of today's lecture. So what Darwin understood, and I actually find this very remarkable, he understood this in part because he studied variation in many different venues. So he has an entire chapter on variation under domestication, where he basically looks at farm animals, okay? And what he finds, which is remarkably insightful given the fact that he has no idea there's even a thing called a gene, right? Is that the whole organization of the organism is so tied together during growth and development that when small variations in one part occur, other parts become modified. So the statement is the following: you can't change everything in every way. If you change one thing a little bit, another thing will get dragged along. So a good example is, as hominids have gotten bigger, their heads have also gotten bigger. Is it because we have selection to get taller and our heads get bigger? Or when you build a taller human, does their head just get bigger for some reason, right? So his idea was that there are trade-offs, okay? So evolution can't have it all. There's my title. All right, so this is Darwin's realization in 1859. So let's talk about trade-offs. That's what we're going to talk about today. And when you go to the car dealerships on I-74 out there, you make a decision. You make a trade-off. Do you want to get a Prius that gets 50 miles per gallon, right? You can go to Chicago on like 2.5 gallons of gas. It's going to take you a while because it takes 10 seconds to get to 60 miles an hour in the Prius, right? Or you could spend a fair bit more money. You could get 21 miles per gallon for $190,000 in your Porsche 911 Turbo, but you can get to 60 miles an hour in just 2.8 seconds, right? So there's a trade-off. You want to go fast, you want to get there quickly, you got to spend gas, okay? And so this is actually an intrinsic constraint on the way we build an internal combustion engine. If I want an engine with a ton of power, I make, a 4-liter V8, right, that can, boom, make that car go really fast. But it's going to burn a lot of gas because the engine has a big displacement. Okay. So it's remarkable, actually, that this idea of trade-offs is now pervasive in not only biology and engineering, but also economics. And so I'm married to an economist, so I sometimes try to think about these ideas to get her interested in what I'm doing. It sometimes works, actually. Okay, so here's an idea which actually comes from an economist named Pareto. In the 19th century, he was asking the following question in an economics context, but I'm going to do it in a car context. Where he's asking, okay, so given some trade-off, what's the best car I can build? So here's a simulation from some people where they're plotting the amount of fuel your car uses, okay, liters per 100 kilometers traveled, versus how long it takes you to travel that distance, okay? So this car down here in this red point drives a lot faster, right? Because it doesn't take very long, right? The time is short. But it uses a ton of fuel, okay? That's the Porsche. This car is the Prius. It takes a really long time to get where it's going, but it doesn't use much fuel, okay? This car right here, don't buy that car, right? Nobody should buy that car. And the reason that you shouldn't buy that car is because it's not Pareto optimal. What is Pareto optimality? It's all the cars that are marked by these red points, right? Why shouldn't I buy this car right here? Because I can build a car that's better in both its efficiency and its speed, right? But once I'm on this Pareto front, if you will, or once I reach a Pareto optimal solution to this trade-off, if I want to improve my efficiency, I have to give up some travel time. If I want to improve my travel time, I have to give up some efficiency. Okay? So this is something that happens in manufacturing. This is something that happens in economic decision making. And the point is very simple. There isn't a best car. There's a set of Pareto optimal cars. Those are these red points. Any one of those is optimal. And if you want to choose between those red points, you make a subjective decision. I don't want to use a lot of gas, or I want to get to 60 miles an hour in 2.8 seconds. Right? That's the trade-off. Okay. So what I'm going to argue for you is that evolution, and I'm going to show you a very explicit example from my own lab, that evolution is subjected to this very same type of constraint. Evolution often happens on Pareto fronts, meaning when evolution wants to vary one thing, another thing changes, and that has impacts on those organisms' fitness. Okay. Is everybody with me on a Pareto front, on a trade-off? I think it's a very intuitive and nice idea. It actually turns out to be quite relevant. Okay. The red points are Pareto optimal. Okay, so evolved machines. So this is one thing that my physics colleagues have a hard time appreciating early on when they talk about biology. In physics, we don't think about things having function. We think about an electron, it's got some laws, the electron is subjected to those laws. In biology, we're studying systems that do jobs, right? And so when we study systems that do jobs, they're subject to these functional constraints. So I'm just going to give you a few short vignettes before I launch into like a detailed exposition of my own work about evolved machines, which we are a very nice example, I would argue, at all scales are subjected to these trade-offs. And I'm going to look at an example at the molecular level, at the bacterial level, which is the one that I know and love the best, and then even in a mouse. Okay? So to give you an idea of scale, I mean, we don't generally think about these scales in our everyday lives, but the protein I'm going to talk about is about 10,000 times smaller than the width of a human hair. So a human hair is a nice ruler because you can hold it up and sort of see it in the light. That's about 100 microns. Bacteria are about 1 micron, so they're 100 times smaller than the width of that hair. Mice, you know how big mice are, so you can see the mouse. Okay. So let's go to the world's most popular protein, can't have it all. Okay, so the world's most popular protein. The protein that's most prevalent on the face of the earth is Rubisco. How many people have heard of Rubisco? All right, excellent. There's some photosynthesis experts here. So Rubisco is responsible, this is a cycle here that happens in the metabolism of photosynthetic organisms. It's right here, and it's the protein that takes carbon dioxide out of the air, we should be thankful for Rubisco, and incorporates it into organic matter. Okay? So this protein is catalyzing, that is affecting a very important chemical reaction, right? It's dealing with at least some of the CO2 that we're pumping into the atmosphere. So what one would really like to do is to build the best Rubisco possible, right? The one that does it the fastest and the best. So my friend Tzvi Tzlusty, who's an Israeli guy now working in Korea, an incredibly sharp theorist, sat down and started pulling data off the internet, and he realized that Rubisco is subject to a trade-off. And here is that trade-off. So he took all the Rubiscos he could find, and he looked at their biochemical parameters. So he asked, how fast does Rubisco get this functional job done? How fast can it bring CO2 into the plant and incorporate it? That's this bottom axis. Okay, that's the speed. Then he asked, how high is the affinity of these enzymes? Higher affinity means it can operate at lower concentrations of CO2. So when the plant doesn't have much CO2 around, if the affinity is high, the system can still operate. Okay? So affinity is increasing in this direction on this axis. Sorry, that's the way we do things. And so the best enzyme is actually that blue point. That would be the highest affinity, highest speed Rubisco you could imagine building. If you look at all the evolved solutions to Rubisco, they actually lie on this line. That's a Pareto frontier. I can't build, well, okay, that's an open question, but evolution has not built this enzyme for us. And so this is an example of a constraint where as I increase the speed, I decrease the affinity of that enzyme. And so my Rubisco kind of gets worse as its speed of this reaction increases. So Rubisco can't have it all in the sense that if you want to incorporate it fast, you need more CO2 to do it. If you want to incorporate it slow, you can do it with less CO2. So that's an example at the molecular level. A natural question then to ask Svi is why, and he doesn't have a good answer for you yet, but this paper is only about 5 years old, so give him another 20 years and maybe he can answer that. A second example of a trade-off comes from the microbial world. So this is the one where I spend a lot of my time. And in the microbial world, we have microbes like bacteria. So in the upper right-hand corner, there's a bacteria with a circular genome. And the bacteria actually turn out to get the flu just like you and I. But sometimes when they get the flu, it's really bad and it's fatal. And so what happens is they get viral infections, but we don't call it a virus when it infects a bacteria. We call it a phage. Okay, so a bacteriophage is a virus that infects only bacteria but not humans. So here's the way the phage works. Phage, just like the viruses that infect us, just like flu, just like HIV, just like hepatitis, it can't replicate itself. It needs the cell to do its replication for it. Okay? By the way, you guys can stop me and ask me questions, but if there are a billion questions, I might keep going. Okay, so don't hesitate to stop me. So the phage attaches itself to the cell of the bacteria. This is the phage, this is the bacteria. Here's actually a false color micrograph of a phage. These things are tiny, they're 100 nanometers. They're 10 times smaller than the bacteria itself. They attach to the bacteria and then they inject their DNA into the bacteria, boom, like a little needle. Okay? Then that DNA goes into the bacteria. It takes all the machinery that the bacteria uses to make proteins, and instead of making more bacteria, it makes more virus. This is the same thing that happens when you get a cold sore, for example. Okay? So what the phage is doing is it's using the bacterial machinery to build more of itself. So now the phage has a problem, because what it's doing is it's infecting a host, right? And if it makes that host sick, that host will do a worse job of making more viruses, right? So if it compromises the ability of the host to make more viruses, then it's actually hurting itself, okay? And so the phage actually also experiences a trade-off, and that trade-off is shown here. So if you look at how much the phage damages the host cell, I won't go into how that's measured, but we can measure that. That's increasing to the right. Okay, so this means when a phage injects its DNA into the host cell, it makes that cell unhealthy. Okay? Here is the number of phage that get produced by the infection. So the DNA goes in, the phage uses the machinery in the bacteria to make as many phage as possible, and then they just explode out of the cell and they kill the cell. That's what's shown here. Okay, sorry, I should have pointed that out. Okay, so they explode, and what the phage wants is to produce many, many, many more phage, right? So here's the number of phage that's being produced by the infection, and here's how sick the host cell gets when the phage infects it. And so what this is showing you is that the more the phage hurts its host, the worse that host is at creating more phage. Okay, so so this is sort of a very nice example again of a Pareto front or a trade-off in the life cycle of the phage. All right.
It doesn't seem that there's. Well, I'm sorry, I have a pretty subjective question. It doesn't seem like it would be beneficial to cause more damage.
Yeah.
But that's kind of like a.
Yeah. So this is a hard thing to deal with, right? So why are some viruses evolving to be very, very bad? So a good example would be Ebola. Ebola doesn't do well for humans, right? It's nearly 100% fatal.
Does the host also fight against the virus and kill it off?
Right. So this is generally the argument that's made. So the argument that's made is that the virulence has to increase in the phage to decrease the ability's host to shut down the infection when it starts, right? So if the phage becomes too weak in its inhibition of the host, then the host may just evade the reproduction of the phage altogether. And there are many, many mechanisms by which the host can actually inhibit the phage from reproducing at all once it gets infected. The most popular one, which you're hearing about every day in the New York Times right now, is CRISPR. CRISPR is actually an adaptive immune system of bacteria to phage infection. And so the phage, if they get a little lazy and they go too far up this direction, then basically the bacteria will shut down their infection in some cases. Right. So see, they have to do some damage, but you don't want to do too much damage, right? So what these guys did was they did selection under very specific conditions where they were able to get some that did a lot of damage and some that did a little damage, and then they see this negative correlation. Dental constraints. So it turns out that the teeth can't be any shape they want. And so there's some work by a group in Sweden where they've been cutting apart mouse heads and doing small-scale tomography with x-rays on all their teeth and studying their molars in particular. And so if you look at a mouse, I'm not an expert on mouse molars, but I've read these papers in the last few years. They're sort of interesting. So the molars, there are basically 3 molars in a mouse, M1, M2, and M3. And what these guys have been doing is looking at across evolutionary lineages, so going from one mouse to the next and looking at the shape of the molars, and then also understanding whether they're herbivores, carnivores, et cetera, omnivores. And what they've shown using some really horrifyingly difficult experiments is that these molars actually develop sequentially. So M1 develops first, and then M2 develops, and then M3 develops. And the way that it works is that when M1 is developing, it stops the development of M2 up to some point, and then that inhibition is released and M2 starts growing. And then after M2 grows for some period of time, it stops inhibiting the development of M3, and then M3 grows. Okay? So what that means is that you can't, if you want M3 to be really big, then M2 also has to be really big because M2 is responsible for regulating when M3 starts growing. And the whole process is constrained by the amount of time it takes to build a mouse head, basically. Okay? So the really sort of shocking result, I think, from all of this study is this plot. So this is showing the ratio of M2 to M1. That is, if you have a big M2, an M2 that's about the same size as M1, you're up here. Okay? So that means you're an herbivore. These 2 teeth are basically the same size. If these 2 teeth are basically the same size, what you find is that M3 is also about the same size. Okay? On the other hand, if you want to make M3 really small, you also have to make M2 smaller relative to M1 because of this sequential cascade of the tooth development process. So here's another Pareto frontier. So now we've gone from a Rubisco enzyme to the size of teeth inside a mouse head. And what we're seeing is that you can't just make, say, M3 arbitrarily large or M2 arbitrarily small because the development of each tooth is sequentially constrained by the development of the next one. Okay, so these are all constraints on the developmental dynamics which lead to constraints on the shape of these teeth. And the really surprising thing is that along this Pareto frontier, they see that there are different diets. The herbivores have big, broad molars, and those big, broad molars are good for cutting up plant material, but bad for ripping into flesh, right? Then you have the omnivores that have, a little sharper molar that's good for cutting up meat, and then you have a faunivore, right? And that's a carnivore. Okay, so this Pareto frontier describes evolution along a diet axis, which you can then see in terms of the shape and size of these teeth, and these folks understand the underlying molecular mechanism of the whole process. It's really a remarkable discovery. Okay, any questions so far?
So would the correlation between the size of herbivore, omnivores, or faunivores be correlated to obviously it's correlated to the diet, but
Yeah.
would there be any, what information would there be for the bacteria capable of digesting those foods?
Oh, I don't know. It's a tough connection, actually. You mean in their guts?
Yeah.
So to the shape of the teeth?
So how would the bacteria in the gut capable of consuming those diets affect the evolution of the teeth?
I have no idea. It's a good question. I mean, I would expect that the bacteria in the gut of the herbivore are different from the bacteria of the gut of the carnivore, but I'm not sure there's a causal arrow towards the teeth. If there were, we should talk, because it's probably a very interesting paper to write. But I think that might be an onerous experiment to try to convince somebody. Yes. Yes, please.
That diagram explains perfectly why my cats have very large front teeth, because they're pure meat eaters. And so I always wondered why they have very
Yeah. So one of the really interesting things is to look at these constraints in one system. So these are all murine species. These are mice, right? There's many different species of mice. And then to understand the molecular mechanisms, right? Because one of Darwin's big results is that we're all related at some level. And so when you have a developmental constraint in a mouse, do you also see it in a cat, right? And so people have done this type of comparative thing which says, okay, I see that, the teeth in the mice have this constraint where I can't arbitrarily make M3 as big as I want without changing the size of M2. Do I see that same pattern in humans, let's say, or in hominids? And in fact, you do. These people have also done that work. And then are the molecular mechanisms the same because they've been transmitted by vertical inheritance through, a billion years of evolution? Yes?
What happens to M3 if you kill M2 very early?
It's a very sharp question. It doesn't develop. Because the question was, what happens to M3 if you kill M2 early in the development? So you're asking for what they call an ablation experiment. And my understanding, I am not an expert in this field, but my understanding is that M3 will not develop because there are necessary chemicals that diffuse towards M3 to cause its development. But we should go read those papers more carefully together, maybe. Anybody else? Okay, so just a brief summary before I tell you a detailed story about this type of Pareto optimality showing up in my own group. So trade-offs constrain phenotypes. If I have two traits, these could be the size of tooth 1, the size of tooth 2. They could be Rubisco's affinity and its velocity. They're constrained. And one sort of common idea for why, you'll notice that in many of these cases, I didn't tell you why. I didn't tell you why Rubisco was that way. I sort of I can make sort of a crude argument about the phage story, but it's not all that satisfying. In the mouse story, I think we do know why. But the sort of generic idea in biology for why is that genes that phenotypes are polygenic, that one gene doesn't control one phenotype. So a classic example is eye color. How many genes determine eye color? Something like 4 or 5 at least. And so if you make one change to one gene, it affects multiple different traits. Eye color and, say, hair color can be correlated, right? And so when you have a specific gene and you change that gene, it may affect this trait number 1, but then it also has what's called a pleiotropic effect on trait number 2. And this intrinsic sort of multifactorial interaction between genes and phenotypes causes these constraints. That's the qualitative argument, at least. And so we set out, when I first came to Urbana, to think about this type of problem in what is probably will come as a surprise to you, the best studied biological system on Earth. And that is the ability of E. coli to swim up a gradient of food. This is your tax dollars hard at work, in case you were wondering, right? It's been worth it, I will tell you. Okay, I've argued an entire semester in my undergrad special topics course that it's worth it. So a model system is motility in E. coli. And E. coli is this bacterium that we've been studying for 100 years. And one of the processes that it does is it swims toward food. So the white side of the slide is like a sugar cube or something. Think about it that way. And E. coli wants to go towards that sugar cube. And it can only do 2 things. It can either turn around, spin in circles, or it can swim straight. Those are the only 2 things it can do. It doesn't have eyes, right? It has little sensors that can smell chemicals effectively. But imagine your eyes are closed and you can only feel the chemicals that are right next to you. How do you go find that food source? So what it does is it swims straight for a little bit and it measures. It says, is the environment getting better? Am I getting more food? Yes, keep swimming. Okay? And then at some point it just spins in circles again. and swims in another random direction. And if the food continues to get better, it keeps going longer, right? And so what this results in is a random process where sometimes it swims the wrong direction, but when it swims the right direction, it goes a little further. And that causes it slowly to drift towards the food source on the right side of the slide through a process that in physics we've studied for a long time called a biased random walk. And so I'll show you, this is a thing that we know a lot about. Let me show you a YouTube video. YouTube always makes it real, right? So these are some E. coli in the lab of a guy named Howard Berg at Harvard University, who was a physicist, first at Boulder, who had a remarkable insight. Basically in the 70s, he noticed that the bacterial geneticist had been studying this process, and he thought it was a good model system for understanding fundamental questions in biology. And he did a series of papers over about 40 years, which were really remarkable in their insight into biology and their use of physics. I think it's the best example of physics actually contributing to biology. There's many examples where we've maybe not contributed so much, but this is one where we've done well. How do I do this? Okay. There we go. So PJ Masks will come on right after because my kid. Anyway, we'll just watch this short video. So what you see is where's my pointer? What you see is, this is an E. coli cell. And these are its little flagella. So it's got these long fibrous things that spin in a corkscrew and push it through the fluid. Okay, so E. coli can't coast. That's a whole other story. It has to push itself through the fluid. So here it is swimming around. I'll show you some more videos. You can see those corkscrews sort of spin around, and they cause it to get propelled through the liquid. And that causes this biased random walk that I was just talking about. Okay, so hello. I'll show you more videos, don't worry. So you can see this in the lab, and this is a video from my lab, which we took with a $50 webcam we bought on Amazon and some LEDs. And what you're seeing is an agar plate. It's about this big, and it's a gelatinous material, kind of like really soft jello, okay? And it's got a ton of food that E. coli loves in it. What my student did is you can see a little sort of scar here, put a few bacteria there, just like 1 million, which is not many. And then what happens is a remarkable thing. So I'll play the video once and then I'll explain what's happening. Looks like nothing's happening, and then there's a supernova. Boom. A bacterial supernova, if you will. So what's happening is that at that center position, you have some bacteria. They're in an ocean of delicious food. So what do? They start eating that food. And when they eat that food, that means they reduce the concentration of food at the center of the plate, but right next door to them, there's more food. So then they start swimming out towards that food and eating more food. Then they start swimming out and eating more food and swimming out and so on. And then you get this beautiful concentric ring. So the white part is all bacteria. There's something like 10 billion bacteria in this tiny little plate. We can count them. Okay? And so this outermost front is what we would call a traveling wave of bacteria, which is driven by, critically, 2 processes. I'd like to say I knew this experiment was going to come out the way it did, but of course I didn't. 2 processes are the swimming that I just described to you and their growth, their ability to eat the food. Okay, so what we thought was, let's just pick the fast bacteria and see if they go faster. This is kind of how science goes when you don't really know what you're doing. The department chair is not here, right? So I can say whatever I want. So what my student did is, the bacteria expand. This takes 12 hours, by the way. And then he goes and he picks the fast ones, and he puts them in there again. He starts them again. Then he goes and picks the fast ones 12 hours later. He puts them in again. Picks the fast ones, puts them in again. And what's happening? All those movies happened on a time scale of 12 hours. They're getting faster and faster and faster. Of course, the poor guy has to come in every 12 hours, which doesn't sound that bad, but it's kind of onerous. And so what he's doing is he's picking the fast ones and starting the next one, picking the fast one, starting the next one, and so on. And the bacteria are speeding up. And so then we can do some image analysis, and we can plot how fast the bacteria are going as a function of how many times we've done this selection process. Okay? And so they get about twice as fast. Okay, that's cool, but the question is then why and how? And so ignore the Greek letters. there's basically three things going on in this system that I've sort of described for you already. One is diffusion. The bacteria just swim around randomly. If there's no food around, they just keep swimming and moving randomly. The second thing is this process that I just described earlier, where if the food is better in one direction, they tend to go that way. That's called chemotaxis. It's taxis towards a chemical attractant. The third process is growth. That's just one cell makes 2, makes 4, makes 8, and so on. Okay? And so these three processes you can write down in terms of an equation. An equation's not important, but the equation lets you ask the following things. If I'm a bacteria and I want to end up at the outermost front of this supernova, what do I have to do? What's the best thing for me to do? Should I speed up my growth? Because growth is involved in this process, right? They have to eat the food to make the gradient, to swim up the gradient, to get out to the edge. Okay? But I could also get really good at this. I could get really good at beating my neighbors to that front. And so this is really, in the lab, a process of natural selection. And this plot that I'm showing you is evolution that's happening in a time scale of 5 days in our lab.
It's interesting that they start going down after the longer the repetitions go.
This is an artifact, I believe, if that's what you're referring to. Yeah, I don't think they actually go down, but
Okay.
yeah. There's issues with the measurement that cause them. Yeah. Any other questions? Okay. Yes.
Yeah, I'm just wondering. When you start out, you think all the E. coli was the same? So
we made them all the same.
Are you just removing the ones that weren't there originally, and also sort of in a natural E. coli constant population? Would the optimum thing be an ecosystem with different species?
Yeah. So what we do is we can do a very easy trick to make sure they're all the same at the beginning.
Okay.
So we initiate the entire experiment from one cell, and that's not hard. So then they all have to be the same, roughly. But here's a crazy thing about bacteria. You're not used to thinking about populations of 1 billion. So when we grow one test tube overnight, we have 1 billion cells. And in that test tube, there exists probably, on average, 1 copy of every mutation in the entire genome of E. coli, which is 4.6 million letters of DNA. So you can find a lot of variation fast if you're a bacteria. This is impossible in humans, right? Because our populations are small. Okay, so we can do some math, we do some simulations. I find some students that are smarter than me, and they integrate these equations, and then they tell me the following result. The heat map is telling you how fast is this supernova expanding. So the yellow is really fast, and the green is slow. And the bottom is telling me how fast the bacteria split and divide and grow. And this is telling me how fast they swim when they do that straight swimming that I described. Okay? So what this tells me is that if I want to migrate if I want to do a fast bacterial supernova, I should both, this is key, grow fast and swim fast. Everybody with me? This is important for the next 10 minutes. Okay? All right, so you got to grow fast, you got to swim fast. And so by growth, I mean the population growth. So I mean, how fast does one cell become 2? That's the growth rate. The faster one cell becomes 2, the faster they eat up the food, the faster they make that spatial gradient in the food supply, and the faster they push that supernova out to the edge. Okay, so the prediction, although it's based on some perhaps dubious assumptions about what's possible, is that you should migrate fast by increasing both how fast you swim and how fast you grow. Okay? So then we just go ask, how fast are they growing? And they're growing slower the faster they migrate. Okay, so what I'm showing you here is how fast they grow, that is how fast one cell becomes 2, as a function of the round of selection. So these are the very fast migrating strains after they've been selected for 15 rounds. Okay, so these are the ones that are migrating almost twice as fast. They're making these supernova twice as fast as these ones, but they're actually growing slower for some reason. So at this point, we think, okay, now we've got something potentially interesting. So this is a population level measurement, but what we really need to see is what the little guys are doing at the individual cell level. So then we have to get a little bit more sophisticated and build a microfluidic device, which is a small liquid chamber where we trap a couple of cells. So here's a microfluidic device. This is 200 microns across. The whole red circle is 200 microns across, which is 2 human hairs, roughly. And in that device, there's two cells. These are sisters because we watch them divide. Okay, so we call them sisters. And we're imaging them with a microscope. It sounds harder than it is. It's not that hard. And so there they are swimming around. Cute, huh? So you can see them sort of reorienting. They do these little, they call them twiddles or tumbles. And then they swim in straight directions. Okay? So the way we set up this measurement is we can put hundreds and hundreds and hundreds of cells in these things and get massive statistics for how they're swimming. And then we can take cells from before the evolution process and after the evolution process and make comparisons. So we write a bunch of code and we extract their trajectories from these videos, and we do a bunch of analysis on this. And then we say, okay, I'm going to compare how fast you were swimming before we selected you for swimming and growth, and how fast you were swimming after. And this is what we get. What we get is that the swimming speed increases with selection. So this white curve is the cells before we selected them to do this very fast migration process, and then the red and the orange is afterwards. So they're swimming 50% faster. Okay. So here's the plot that I just showed you. What our model told us was you should swim fast and grow fast if you want to do this migration process very fast. When we do the experiment, when we measure the strains after we perform selection, what's happening is they're swimming fast, but they're growing slow. That's a Porsche, right? So this is the inefficient, if you will, fast-swimming bacterial lineage. Okay? And the way we did this experiment was we gave them a lot of food. So they're growing really fast in this situation. Okay? And so then we thought, let's try this again, but let's try it in a situation where they don't have a lot of food. Let's try this again in a situation where, oh, excuse me. I'm going to tell you another brief story first, although I might skip it. 45 minutes? Should I?
No, you can go.
Keep going? Okay. Okay. Okay, I'll tell you the genetics then briefly. So then what we asked was, how is this being done at the genetic level? So first we submitted our paper, and then the biologist said, yeah, we don't care until you tell us what the genes are. And so then what we did was we did a bunch of sequencing. You can ignore this slide, basically. And we found that in every run of this experiment, we were seeing very similar mutations. And those mutations were doing something in the genome that we could tell because we know so much about E. coli. So in the genome of E. coli, there's a protein which has an acronym name which means nothing and should not be interpreted. And this gene, what it does is it turns off the genes that control swimming behavior in the cell. And the mutation that we saw, so this blunted arrow means turn off, and these are the genes for getting the cell to swim. Okay? The mutation that we saw broke that gene. We can tell it broke that gene because we know the genetic code. Okay? So that's an interesting result. What it does is it breaks the repressor. So this thing is repressing motility. You break that repressor and you get strains that swim faster. So then to really convince the editor, the biologists, you have to actually take just that mutation, one letter change in the entire genome of E. coli, put it in the genome with a technology that's basically the same as CRISPR, and then ask what happens to those cells. And so when we do that, we find a year later that, that it both swims fast and grows slow. One letter change in the whole genome. Okay? So we put this single mutation in the genome of E. coli, and it causes the cells to grow slower and swim faster. That's exactly what I was telling you before, was the reason for these trade-offs, right? The reason for these trade-offs is because a single mutation affects two things at the same time. So it's not like, you know, there could be another situation. It's not like there's a bunch of mutations that make you swim faster and then a bunch that make you grow slower. There's one mutation that makes you swim faster and grow slower. And that's what a geneticist would call pleiotropy. In physics, I guess we would call that like a higher order effect.
What was that? Biotropy?
Pleiotropy. Yes, confusing, but okay. All right, so then we do the whole experiment again, except for we do it in a situation where the cells don't get a lot of food. So, and they, again, are able to migrate faster as we select them to go faster and faster. Okay? Yes?
Would that be the reason why, since you have a lot of
Right. So the question of cell size is a good one. So one question, I'm going to answer your question with a different answer than you might want, but it made me think of something that I should say. One question is why? Why do cells who swim faster, grow slower? And one hypothesis that we had is that they actually get smaller. And if they're smaller, they experience less resistance as they push themselves through the fluid. So we spent a while looking at that. It doesn't really seem to be true. I had a really good undergrad. She did an amazing job, but we didn't.
The use of energy?
Yes. So the other question is the use of energy. So the spinning of the flagella takes energy. This is actually probably the most likely hypothesis for this trade-off. And so if you grow a little bit slower, you have a little less energy. Or if you grow a little bit slower, you have more energy to spin the flagella, and then you swim faster. This requires other measurements of the energy balance in the cell, which can be done, but we have not done yet. Let me finish the story, and then you can ask as many questions as you like. Well, maybe not as many as you like. So we redo this experiment in in very little food where the cells are starving. They still are able to make this migration happen faster. And then we do the same measurements. We measure how fast are you swimming and how fast are you growing after we select you for this migration process in a little bit of food. And what we find is that they actually slow down their swimming in this case. So these are the cells before we perform the selection, and these are the cells after we perform the selection. So rather than swimming at 20 micron per second, they're swimming at something like 10. And if we look at how fast one cell becomes 2, at how fast they're dividing, they've increased that by almost a factor of 3. So here's the cells before we perform selection, here's the cells after. Okay? So this is the opposite answer, right? So now if I go look at this space of swimming speed, sorry, I should have the words here, and growth rate, what I find is that they started up here and they go down and to the right. They grow faster, but they swim slower. Okay? That's the Prius. So these are the cells that, with a little bit of food, they can go a really long way, but they can't get there as fast, right? They can't get there as fast. And so then, the geneticists are still not happy. We have to do the genetics again, and I'll tell you that story briefly. It actually turns out to be interesting. So the summary of these two is that when you're well-fed, you evolve the Porsche strategy, and when you're poorly fed, you evolve the Prius strategy. And what is this curve here? This is a Pareto front that the cells are constrained to, just like we saw before, right? What I really want is a cell up here. Again, this is how fast you grow and how fast you swim. A cell up here, all my simulations are telling me would be the best possible cell. Why don't we see that? There's some intrinsic constraint in the system, this engineered evolved system, which says you can't do both. You can't have it all. Okay, so then we go do the genetics again. Again, we find similar mutations over and over again in this starvation condition environment. Again, we find that mutation is breaking an element in the genome which is repressing something else. So it's turning things off. So this is a gene that controls not the motility, but the metabolism in the cells. So this is a gene that controls how the cells take up the nutrient that we selected them in. And the mutation we observe is breaking that negative interaction. So this gene turns something off. There's a mutation that breaks its ability to turn that thing off. Okay? So we're breaking a repressor again. So this, in the end, I was initially grumpy when the editor said, go do the genetics. I was like, I don't want to do the genetics. It's too much work. We did the genetics, and we actually learned it was worth it. And the reason it was worth it is because we see this very similar mechanistic basis in both cases. We see the disruption of a negative regulatory element. That's probably too jargony. We see this breaking of a switch that turns something off, which is an easy thing for a mutation to do. If you have something that's turning something off, and you just change a little bit, it's unable to then continue to turn that thing off. And so then, again, remarkably, yes?
Did you ever try the go backwards experiment where you change the concentration of food? You run your 15 rounds with high energy, and then you run your 15 rounds with low energy, but you take as your starting round for the low energy, the last round of high energy?
No, we have done crossing experiments where we ask, does the mutation that you evolved in rich medium help you in the starvation condition? And the answer is there's like no crossover.
But you never see if
So we've tested the Porsches in the Prius conditions, and they're not faster.
they're not.
No, yeah.
You can't see if a Porsche goes back to a Prius.
No, we haven't tried that. We have done the selection where we alternate starvation, fast, then they get better at both with different mutations.
Oh, wow.
So the story I'm telling you is clean, but I've cherry-picked it a little bit. Okay, so anyway, this mutation actually does exactly the same thing. It's pleiotropic in the sense that one mutation, just one letter change out of 4.6 million, causes them to swim slower and grow faster at the same time. So again, there's not a bunch of mutations a few that make you grow faster and a few that make you swim slower. You change one thing, and it affects both at the same time. Okay, and that's why there is this Pareto front in the phenotypes that we see. So the interesting thing, and actually I have several colleagues, especially one in Germany, who's done some really remarkable work showing that you have this Pareto front, and in both cases, what you're doing to adapt in the lab conditions that we have, at least, is breaking this thing that turns something off in the genome. And so what we found collectively as a community is that when you perform strong evolutionary selection on short timescales in the lab, the easiest thing for the organism to do is go around just taking a hammer to stuff. Let's just break stuff because it's fast and easy, right? They're not evolving a slightly better enzyme, which is really hard and slow. They break a bunch of stuff, they see what works, and then they adapt. And so that's, I think, one of the sort of compelling stories that comes out of this work. So in summary, I just want to say, we've talked about these car designs that you make a decision about when you buy a car, and you're subjected to this constraint, which is intrinsic in the way the system is built. You have an internal combustion engine, you can't drive fast and do it efficiently. And in the microbial world, we see a similar type of, it's a loose analogy at least, similar type of Pareto constraint. So it may seem like I did all the work, but I actually did very little of the work. These three people were the primary drivers. So Dave and Harry are PhD students in the physics department here. And they've taken extremely well to doing, in some cases, quite hardcore biology, especially Dave. Diane was an incredibly talented undergrad, the daughter of a UIUC professor. And she was really important in doing this work, and she's now a PhD student at UChicago in the biophysics department. And this is the rest of my brave set of students who seem to be willing to spend a lot of time with me. So thank you, and I'll take any questions that you have.
We have time for some questions. Maybe here.
I was wondering,
Lady first.
How concerning is it if the Chinese scientists and others do that use CRISPR to insert or change the DNA of a child?
I mean, it's concerning. It's not a scientific question, though, right?
We know a lot about E. coli, but we don't know a lot about E. coli as we start to manipulate.
Okay, I think I have a reasonable way to. So here's what I would say. Even in E. coli, it's very, very hard to understand what a mutation is going to do. And I did this. We did this experiment in my lab. We didn't know how it was going to come out, right? So I think one thing that's a bit understated in the popular coverage of CRISPR is how hard it is to know what a specific genetic change will do to an organism. And there are cases where that's not the case. Cystic fibrosis, sickle cell anemia, those are what we call Mendelian traits. They're very easy to understand. If you want something more complicated like Alzheimer's, it's not going to work, I don't think. And I think the reason is that, as my postdoc advisor used to say, DNA is overrated. And the organism is not simply encoded in a very simple way in the genome. It may be encoded in there, but that encoding is very complicated. And it may also depend very strongly on non-genetic factors. So you're hearing a lot about epigenetics. That's one thing. But there's a whole physiology of this organism which depends on many variables other than the exact set of letters that you see in the genome. So I think a little bit what's happening with CRISPR is that we can now sequence and manipulate DNA very well, and we understand that level. Okay, so we can control that level better than we understand that level. And so I think that's one of the things that's really scary. In addition to all of the ethical things about, you know, eugenics, that's a whole other horrifying situation. To be fair, the main people who have been pushing CRISPR in this country have been very vocal about not editing human genomes, like just an international moratorium. But
Up there.
Yeah, on the penultimate slide, before the credits anyway, you have the yellow arrow that said selection.
yeah,
Is that, I was wondering, is that an evolutionary equivalent to the Wolf effect? Economics, where you're actually moving the Pareto frontier? So, for example, if you were to do the graph with cars
yeah,
in 1920,
yeah,
you'd get a different Pareto frontier.
Yeah.
And so over time,
yeah.
so is that the arrow of evolution?
That's not what I mean here, but that's an insightful statement. What I mean here is that selection is pushing the organism to this direction because it gives you the fastest migration in my experiment. What you're referring to is something a little bit deeper, I think, which is, you know, as the engineering for internal combustion engines gets better and better, you get more efficiency at the same amount of power, right? And so, because the cars are getting better. And so that actually migrates the Pareto front. I don't know of an example of that in biology, but it would be a very interesting thing to think about.
But evolving those better enzymes, that
Yeah.
you mentioned, the slow process,
Yeah.
could move the whole curve.
Yes, unless there's a fundamental physical constraint or chemical constraint, right? Like, unless you're up against the amount of energy per ATP molecule, which is not going to change, right? So fundamentally, for example, in solar panels, they've been getting better and better and better. We know there's a quantum limit. Quantum mechanics will not let you get better than, do you know what the limit is? I don't remember off the top of my head. 40, 50%, whatever it is. At that point, you can stop because the Pareto front will stop moving. It's a good question. Yes?
I just had one kind of related to that, which is, I mean, did you say that you actually have found that optimal E. coli does not exist because in a large population you're sampling every possible genetic variation?
Yeah. So there's a guy in Minnesota that claims they evolved through exactly this Pareto frontier. I don't really believe the paper, but yeah. So one question is, if you just keep going and going and going, is it just that you've only sampled a little bit of genetic variation near where you started, and the system could eventually sort of wrap around and go up there? That's his claim. I can't rule it out in my experiment. I can say on this time scale we don't see it. Yeah.
All right. So maybe one final question.
Is there any particular reason you stretched the bottom numbers on your graph?
It's log scale.
So much more space.
It's log scale.
Okay.
And I did it because it looks better. And I didn't publish this figure for that reason, because it's a bit disingenuous. So this is, you'll notice this is a change of almost 400%, and this is a change of 50%, right? So really the way this thing looks is like that. But nice eye.
Okay, so Seppe will stay around. If you have more questions, you can come down and check.